Right, okay, thanks. Well, it's a great pleasure to be here. Just to correct all the points you've made there, this is who I actually am. So I've trained at Cambridge, London, spent time at Barts. And then the defining moment in my career is I went and did a PhD, actually in my mid-30s, courtesy of the Wellcome Trust. And then shortly after that, I came here and got my first chair at Queens. And I was here from 1999 to 2001, then went to Birmingham, then to Warwick. And if you want to follow me on Twitter and YouTube, there's some details. And here's a trip down memory lane. This is the last time I was here, 15 years ago. You might recognise that young lady there and that man sitting next to her, both in the audience here. And this was my research group in the lab we used to have over on the Royal Victoria Hospital site. In some ways it seems just like yesterday, other ways it seems an awful long time ago. And you can see the city hospital there in the background. But interestingly, part of my research started really when I was parachuted into this land of hypothesis-driven research during my PhD, just a few years before I came to Queen's. And I was kind of parachuted in as a medic into these rather hostile uplands here where you did things like hypothesis-driven research on transcription regulation, and used curiosity, looked at mechanisms and pathogenesis. And this was all rather different from the translational research that medics normally do. And what I found is that actually I was parachuted in there, and the rest of my career I've been sort of beating a path back to where I came from and actually adapting all of the knowledge and know-how and so forth that I've got from here and moving it down onto these perhaps more fertile lowlands of translational research. And so I'm going to take you through a quick overview of many of the things that I've done. Not everything I've done since I left Queen's, because that might take a bit too long. I'm a little bit worried I'm going to cover too much too quickly, but I'll try and keep up and keep the pace up as we go through. So, to start with the excitement of genomics, and this is something we're talking about over lunch, it's still very, very exciting, just the idea that you could sequence bacterial genomes. And I was here at the outset of this revolution when we started sequencing bacterial genomes, and in fact that paper there in Nature, Parker et al., my name is on it with a Queen's affiliation. And it really was enough to make you feel poetic. You know, there's a quote here from a Keats poem on first looking at Chapman's Homer. This was the time in which organisms that we'd seen down a microscope on a gram stain or whatever, suddenly we could see them in all their glory on our computers. We could see their inner workings of humankind's most fearful adversaries, the organisms that cause the Black Death or the White Plague. All of this suddenly became available to us, and it was a very exhilarating time to be involved. Sheila, of course, was also involved because she did the Bacteroides, involved in the first Bacteroides genome a year or two later. So it was a very exciting time, but the excitement never stopped, because fast forward a few more years and we have high throughput sequencing. So people who play conference bingo, you can always tick this one off. You always have to put up this slide here, this graph showing the cost of sequencing. And you can see that the cost of sequencing came down steadily from those heady years around the turn of the millennium, obeying Moore's law, keeping up with computers. But then something remarkable happened around 2007, where the pace of acceleration changed dramatically. And that's where we got this high throughput sequencing, which allowed us to sequence effectively 1 million times more effectively than back in the old days of Fred Sanger. 1 million times cheaper, 1 million times higher throughput. And also much more accessible in that we got these benchtop instruments. So you could actually do sequencing on a bench this size. You could actually sequence whole genomes, multiple genomes of bacteria in a single experiment. Now I'm going to speak mostly about metagenomics, but I will just pause for one moment to talk a little bit about genomic epidemiology, which is an area which we moved into. And this came out of a clinical need because there was this organism called Acinetobacter baumannii causing problems in the hospital in Birmingham where I was working. So this is a gram-negative bacterium, and it's often multi-drug resistant, and sometimes it can even be moving towards pan resistance, where we get colistin resistance in this organism and hardly anything or nothing to treat it with. Typically associated with wound infections, ventilator-associated pneumonia, bloodstream infections, and just as a strange quirk, it was particularly associated with returning military personnel from the Iraq and Afghanistan conflicts. And that was the case in Europe, but also in North America as well. And at one stage, it was actually given this nickname of the Iraqi Bacter because it was so commonly seen in returning military personnel coming back from Iraq to America. And in both settings, in North America and in the UK, you see transmission from military patients onto civilian patients. So a military patient brings the organism into the hospital and then it spreads onwards. It's quite hard to identify in the clinical lab, and there are some related so-called genomospecies that are very difficult to distinguish phenotypically. And the typical picture that we would see in Birmingham is that and probably I suspect you have the same issues here, is that you'd get serial clonal outbreaks. You get the organism introduced into the hospital environment. It would then be transmitted onwards and sputter on this outbreak for a few generations, and then you get on top of it, it would disappear. Maybe you'd be clear of the organism for a while, and then another one would come in and cause another outbreak. But actually being able to actually tell you've got an outbreak, you could do that using the public health laboratories would actually say, oh yeah, this is a particular outbreak strain of a certain type or whatever. But actually being able to look at the outbreak as it was happening and look in detail at the transmission chains was something that was not possible until we could start doing genome sequencing. So I got involved in this outbreak of MDR, Acinetobacter baumannii, in Birmingham that ran from 2011 to 2012. Now the University Hospital of Birmingham, the NHS Trust, the Queen Elizabeth Hospital, is the major centre where military casualties were repatriated during the Iraq and Afghanistan conflicts. And it's also the site of the Royal Centre of Defence Medicine. And so we were working alongside the microbiologists in the hospital, and they were working alongside some of the military personnel and also all the other doctors in the hospital. First case was was a military patient that came in July 2011, and the reference lab said, yeah, this is a unique pulsar type. You've not seen this before. This is at the beginning of a new outbreak. The outbreak itself was very extensive. It lasted for 80 weeks and involved 60 patients, and those spanned military and civilian patients. And we had just gained control of a MySeq of our own, one of these benchtop sequencing instruments. And so we said, well, let's see if we can genome sequence the isolates from this outbreak as quickly as we can. So we started off, we created a reference genome of the outbreak strain and showed that indeed it was, like Colin Dell had told us, it was a unique strain that we hadn't seen before, that was different from strains implicated in previous outbreaks. And then we went on to look at taking the index case, we then aligned all of the genomes from all the other isolates that we had from the outbreak, and we called out the single nucleotide variants or single nucleotide polymorphisms, the SNPs. So the 1 in a million changes in the genome that were going on over time. So we started off over here with this genotype 1, which was the index patient and close relatives, where these were, as you see, 4 isolates there were indistinguishable. And then as time progressed, we saw the acquisition of additional SNPs as the outbreak went on over time. And these were basically clumped into various major genotypes, which we were able to identify here in these big blocks. But interestingly, where we saw this progression of acquisition of SNPs, we can see here that from genotype 1 to genotype 4, you have to acquire SNP2, then SNP5, then SNP9. All the descendants will now have SNP9. We saw that kind of accumulation over time, so it kind of made sense chronologically. But interestingly, we saw that around some of these major genotypes, we had a cloud of 1 SNP variants. So this genotype 4, you've got all these ones up here that just have 1 SNP different. The numbers are actually the numbers of the patient, and then if we have multiple isolates from the same patient, we put a letter after them. So here you can see we've got 34, 34A, 34E, 34F, but we've got some 34s in here. So within the same patient, we're getting just minor variants as we take the samples, which was an interesting finding. Now, when we superimposed the information we got from genome sequencing onto what we knew from traditional shoe leather epidemiology, the time and the place where the patients were, we got an interesting picture here. So we can see that as the outbreak began, we had these genotypes 1 and 2, mainly focused on a single ward, Ward 1. And this kind of then made sense because we could see that, well, this must be spreading from patient to patient within the same ward. And it's fairly easy to see how that might happen, that patients are in adjacent beds, or maybe the staff are moving from one patient to another very quickly or whatever. That kind of made sense. But as we got to around halfway point in the outbreak, when we started to get to this genotype 4 and onwards, suddenly the Burns unit started to become the predominant place in which we were finding this organism. And that required some explanation. And having the genome sequences, we were able to really be quite clear that, yep, this was ongoing transmission of this serial, of this clonal outbreak, and that we needed to explain it. And the penny dropped when we realised that the common factor, so the problem is on the Burns unit, the patients are all in the individual rooms. So you'd think it would be much harder for it to spread on the Burns unit than on a normal ward where there may be multiple patients in the same ward environment, unless it was spreading. What it turned out was that they were all going to the same Burns operating theatre. So they were going to the same operating theatre to have their Burns debrided. And it turned out that the operating theatre had become contaminated with the organism. And we were able to use that information to do these deep cleans of the Burns theatre, which actually calmed down the outbreak for a while. And then when it flared up again, it did flare up again because someone misused a special bed, didn't clean it properly. We ended up with a second flare up on the Burns unit, and then in that situation we were able to get environmental isolates from that from the Burns theatre and show that they were the same genotype as the clinical isolates we were getting. So this all hung together quite well. And then finally, they did a final deep clean of the Burns unit and actually managed to get the whole thing under control. Now, one of the things you can see also is that when we started doing the sequencing, the outbreak had been running for quite a few weeks. And then we got more and more competent and we started to do more and more runs, and we actually then managed to get to the state where we were keeping up with the outbreak. We were sequencing the isolates as they were coming in, rather than this being, as is often the case in these things, an exercise in freezer genomics. We were doing it in pretty much real time. Now, you could argue, if you're a skeptic, say, well, it's just an outbreak and you would have controlled it anyway. What was the role of genomics? Well, there were many things that genomics actually gave to us. It enabled us to exclude isolates from the outbreak. So we did see some isolates that looked like multi-drug-resistant Acinetobacter, but they turned out to belong to this other species, Acinetobacter pittii. So we could say, well, they're not part of the outbreak, so perhaps you don't have to really concentrate on those. Concentrate on the ones that are in the outbreak, because this is an ongoing problem. We could link the patients via their genotypes, and we could link the patient isolates to the environmental isolates. As I mentioned, we could show within patient clouds of diversity. And another interesting thing, and this will come an issue later in the talk, is we could show that sometimes there are mixed infections, so that we could get more than one genotype of Acinetobacter from the same patient. And that kind of challenges the practice that we just look at colonial morphotypes and assume that everything's the same, which is a common shortcut taken in the lab. And as I say, largely retrospective to start with, but in the end we went to doing from colony to SNP genotype in less than a week. So that was an interesting test case in using genomic epidemiology to actually control an outbreak, help control an outbreak. That was written up in Genome Medicine a couple of years ago, and you can read that if you're interested. Now, around the same time, we also got involved in another outbreak. Some wag decided to call it the sprout break. It was an outbreak of E. coli, a particularly nasty form of E. coli, sugar toxin producing E. coli, that occurred in Germany in May to June of 2011. And there were over 5,000 cases and over 50 deaths. And particularly chilling about this outbreak was that most of those deaths were in individuals who were perfectly well before the outbreak. So Acinetobacter, it would be targeting people who were already rather ill and frail in the hospital. These were young people who've been walking about their business and then they get struck dead. by this organism. It was initially linked to, mistakenly by the epidemiologists to cucumbers, and then they realised actually it's sprouting seeds, bean sprouts. There was a high risk of hemolytic uremic syndrome, the nastiest consequence of this infection in the outbreak. And for some reason, females were particularly at risk. We still don't quite know why. It may be that real men don't eat salad, or maybe there's some other explanation. But we got involved because the outbreak strain was sequenced on a new benchtop instrument called the Iron Torrent by the Chinese Genomics Institute, the BGI, and they released the sequence under a Creative Commons license. And Nick Lohman, who was working in my lab at the time, had just been gearing up to analyse Iron Torrent data because I just won an Iron Torrent in a competition. He sees that and analysed the data, assembled the data, and then using Twitter said to other bioinformaticians around the world, why don't we all have a play with this data and see what we can make of it? And this ended up with us having this crowdsourced analysis using social media of the genome. And again, this culminated in getting a paper in New England Journal of Medicine where we described this combination, what we called open source genomics, where we had benchtop sequencing, open data release, and crowdsourced analyses run by social media. Now, I'm not going to say much about that because I've spoken about it many times in the past. I'm really introducing it to provide context for what we did next. So the interesting thing was that we did all that without actually meeting the German collaborators who were at the heart of the outbreak, the ones having to deal with the frontline issues of that outbreak in Hamburg. And so after we got that paper in New England Journal, we went over to see them in Hamburg, and we had some champagne with them. And here we are, quaffing away a champagne. And I said to Martin Epfelbacher, who's standing next to me here in the picture, I said, what are we going to do next then? Because people just say we got lucky and that we're one-hit wonder, one-trick pony. We should show them we're a smart group of people. What can we do now? And we scratched our heads. I said, well, let's think about unique selling points. I said, our unique selling point is we're pretty good at genomics and bioinformatics. What's your unique selling point? He said, I've got a freezer full of shit. Well, he didn't put it quite that bluntly. I'm just playing to the audience by using such a turn of phrase. He basically said, yes, I've got fecal samples from the outbreak. I've got over 200 fecal samples in my freezer from the outbreak. So I said, well, again, to put it in the vernacular, why don't we just sequence the shit? Why don't we actually take those samples and sequence them and see if we can get to the same place that you got by culture? Now, being in that part of Germany, northern Germany, near to Denmark, it kind of seemed appropriate to start challenging what was going on in bacteriology. So bacteriology was invented in that part of the world, in a sense. So Graham from Denmark invented the Graham stain. Robert Koch showed us that we could actually grow bacteria on solid media with single colonies. And we started off bacteriology like that, and we've been doing the same thing for over a century. I know the virologists, because they can't grow their virus in the same way, they kind of got molecular very quickly, but bacteriologists are stuck in a 19th century groove. And we're dealing with 21st century problems. So I said, why don't we actually try and change things? So instead of doing what we did in the New England Journal paper, where you have your fecal samples, you grow the E. coli, the outbreak strain, in pure culture, and then genome sequence it, can we not go directly from the fecal sample to the genome sequence of the outbreak strain in one fell swoop? So that was the challenge we set ourselves. So I said to Martin, okay, send us some samples then, and we'll have a look and see what we find. We'll just extract the DNA and sequence it. And I was thinking, well, if we get just a few reads that align with the sugar toxin gene to show that basically that organism is there, that will be an interesting output. But in fact, what we found was that the very first sample we looked at, when Nick Lohman aligned the reads from the metagenome against the outbreak strain, he was able to show, this is a coverage plot, we got about 20-fold coverage of the outbreak strain in the metagenome from the fecal sample without optimizing anything, just straight out of the box. And what we also found was that there was this little upswing here in the genome, which is the region that encodes the sugar toxin, encoding phage. And what we assumed that meant was that we were seeing not just those sequences coming out of the E. coli genome, but also sequences coming out of free phage particles that were also present in the fecal sample. So we're kind of getting an insight into the biology, the mechanism of the process of infection just by sequencing these sequences. And this skew we've got here also is indicative this is active growth, this kind of smiling effect. And we detected also we could detect the MLST sequence type directly from the stool sample. So we actually felt pretty cool about that. We thought, wow, we can get 20-fold coverage of a genome just by sequencing the stool sample. Now, JAMA were actually saying that they wanted a special issue on medical genomics, and we contacted them and said, oh, could we publish this kind of stuff? And they spoke to their resident expert on bacterial things, who happens to, I now know, to be David Relman. And David Relman said, well, they're kind of cheating, really, aren't they? Because they know the outbreak strain genome, and they're just aligning the stuff out the stool sample against the outbreak strain genome. That's a bit like doing a jigsaw puzzle, where you just basically take your jigsaw pieces and align them against the lid. You know what it looks like. What you have to do is solve the whole thing without that initial clue. So could we get the outbreak strain genome without knowing in advance what the outbreak strain looked like? What was causing the outbreak even? That was David Relman. I mean, I kind of said that as well to Nick, and Nick had said, oh, don't be so silly. But David Relman had said this too, and therefore we had to fall in line with what the journal editor was saying. So Nick did a couple of, like bioinformaticians do, he did a couple of all-nighters trying to make sense of this problem. And so this is where we started from. He said, okay, let's have a look at sequences that are present in at least two of the outbreak samples. And this is just a graphical representation of the coverage, the number of reads, and the GC content. So he'd done an assembly and ended up with all these contigs. And then he'd done a quick look to assign them to various taxonomic groups. And you can see it's like a zoo in there. There's all sorts of different organisms in there, different taxonomic groups. So to get from that to a single outbreak strain genome seemed a bit of a tricky problem. But Nick's a very bright guy, and he said, well, let's start making some simplifying assumptions. So he said, well, let's assume, so we've got 40 samples here. Let's assume that if this is really an outbreak, that we expect to see representation of the outbreak strain in at least half of the samples that we have in front of us. So any sequences that are not present in half of the samples we have in front of us, we'll throw those away because they can't be the outbreak strain. So he did that, and he ended up with a great simplification here of the taxonomy. So you can see that Enterobacteriales here, these pinkish stuff, they're starting to look more prominent. Still loads of these Clostridiales there. We haven't actually simplified enough down to say anything like a single genome in there. So then Nick thought, well, okay, we've got to solve this some other way. And then he went and thought, well, actually, what we could do is we could subtract what's in normal people. And that might help. Now, luckily, he didn't have to go out onto the streets of Hamburg and ambush 40 healthy Germans and collect their stool samples, because there were ongoing projects, particularly this MetaHit project, where people had done that already. So you could just get out of the databases fecal metagenomes from healthy individuals. So he did that, and he subtracted everything that was in the healthy individuals from this, and then he ended up with very much nearly a single complete genome from the outbreak strain. Now, those of you awake will say, well, actually, if you do that, you haven't actually got the outbreak strain genome, because you will have subtracted all of the core genome of E. coli, because most of normal people have E. coli. So what you end up with is the accessory genome of the outbreak strain. So to add back, he had to add back in the core, and he did some tricks to actually work out what other sequences went with these sequences here as part of the same genome, using depth of coverage and so forth, and ended up with being able to recapitulate the bacterial genome, the outbreak strain genome, from the metagenome, and actually analyse certain parts of the genome, show that certain key operons were there, and we managed to get our paper into JAMA. So we felt very pleased with that approach. Now, around the same time, I was thinking, well, this metagenomics seems to be an interesting approach. It seems to work quite well, actually better than we expected. And one of the key features of the University of Birmingham is they have a bar in the middle of the campus, and every Friday night I used to go to the bar and have a drink or two. I met this guy, Vince Gaffney, who's a rather garrulous Geordie archaeologist who worked on landscape archaeology. And I said to him, why don't we do some ancient DNA stuff? And he said, I don't know about that. I said, yeah, we can sequence DNA. It's kind of cool. And then one day he came back to me and said, okay, yeah, all right, I want you to do something. I thought, oh, he's going to give me a pharaoh's foot or some really exciting thing. He said he wanted me to sequence some mud from the submerged sediments from beneath the sea floor. I said, okay, it doesn't sound very glamorous, but all right, let's have a go. And I then ended up working also with this guy here, Robin Allaby, who looks a bit like kind of Jesus of the wheat field. He's actually a plant molecular biologist who's also developed an interest in ancient DNA. And he is actually working in Warwick. So it just so happened that I ended up going to Warwick. Vince ended up going to the University of Bradford. And so although we'd hatched the project in Birmingham, it came to fruition while we were elsewhere. Now, the issue was I got Vince to explain to me what is it you're really interested in. He said his view was that there was this Neolithic transition, so that was the arrival of farming in the British Isles. Now, any Republicans here, I apologise for saying the British Isles, but if you can tell me another way of saying the name for these islands, I don't know, the Northwest European archipelago, let's call it. The arrival of farming in these islands was a big, significant event in the history of humans, inhabitants here. But according to Vince, he said, well, actually, probably most of the early action here, we can't detect it because there's been sea level rise, and all this area here, so-called Doggerland, or this land that was there between Great Britain and the continent, that's all been submerged progressively over the years. And therefore, that would have obliterated any archaeological evidence of the initial Neolithic transition here. And he managed to get some samples from, he said, well, so the action will be under the water, and we have to turn to submarine archaeology to get the answers. And he managed to get in with some submarine archaeologists who looked at a settlement called Baldner Cliff, just off the coast of the Isle of Wight here. And he said, well, why don't we do sedimentary ancient DNA analysis on it? And at the time he presented this problem to me, this was just bubbling away. There was just a bit of evidence to show that sediments actually store DNA sequences in the absence of macrofossils. So you can get evidence of plant or animal has been in a particular environment, even if there's no bones or any other macrofossils around. And he said, look, what I want you to look for is sheep or goat DNA, because if you could detect sheep or goats, this will be a sign of the Neolithic transition, because these are Middle Eastern animals, and they only belong here because they've been brought here with farming. If you look for pigs, there's wild boar, that are wild relatives. If you look at cows, there's this thing called oryx, which is the wild relative of that. That won't be so helpful, but if you could find sheep or goat DNA. Anyway, so we got these columns, we extracted DNA, the libraries were prepped for high throughput sequencing. This is just showing, I don't know why I put that in actually, that's just showing you more context of the archaeology of the site, and probably the state of the sea, of where the coastline was at the time of the sample. So this Baldner Cliff location was probably several miles inland at the time that the samples were, these sediments were laid down. So we did the work in my lab with our Illumina MySeq, and then I passed it on to Robin and his postdoc to do the analysis. And they basically use a variety of approaches to look for taxonomic signals in the sequences, but they were particularly keen not to fall into the trap of just seeing what you see and believing it. So there's been a lot of fuss about people analysing samples from the New York subway and saying there was plague there. People saying that they detect reeds from duck-billed platypus in their samples when they're nothing to do with Australia. So there are plenty of pitfalls for the unwary when you do these kind of analyses. But they actually managed to to overcome those and came up with some evidence from the non-microbial fraction of animals, mammals, and angiosperms, flowering plants. And the key point was that he found wheat sequences. So we had these four different samples from different time periods, and here's the distribution of the reads that we got, and you can see that in these two top samples here, we've got a lot of samples from the Triticum and Triticii, which is wheat, the genus and the family from which it comes. And actually, wheat, like sheep and goats, is not native to this part of Europe. It was brought by the Neolithic transition, and so this was clear evidence of a Neolithic transition. And it was 2,000 years earlier than was expected. So up until then, people have been saying it was 6,000 years ago that basically these things came here. This was 2,000 years earlier. And given the evidence of direct cultivation of the wheat, this is a bit where I'm not entirely certain how credible it is. But Vince and Robin together said that this probably means that they were trading in wheat and that the wheat had actually been brought there, trading with partners in Europe. So that was an interesting observation. And I still kind of can't quite believe it, but we managed to get that into science. So that got Robin Allaby a promotion to professorship very shortly afterwards. Now, this idea of shock metagenomics was kind of going through my head, and just as I was leaving Birmingham, just about to leave Birmingham, this lady, Helen Donoghue, approached me and said, "Oh, would you be interested in looking at some material that might have TB in it? Can you get sequences out?" And I said, "Well, I don't want to do PCRs because everyone will say that you've got contamination. And it's all, you have to go through a lot of effort to validate your PCRs in the first place, and then show that you haven't got contamination." I said, "Why can't we just sequence the DNA extracted by metagenomics? It seems to be working in other contexts." So this other guy here, this is Mark Spiegelman, who's also involved in the project. He was the one that provided the samples initially. I'll say more about that in a moment. In fact, I saw him just on Friday. We're still keeping in touch and working with him. I said, well, if we did shotgun metagenomics, then we wouldn't have to do this faffing about designing PCRs. We wouldn't have to do hybridization capture, which is the other option. You could make a probe for TB and see if you can fish things out. Why don't we just sequence? Now, you can sit in your armchair and say it's not going to work. Same way as we said that the STEC metagenomics are going to work, because there's going to be high abundance of human DNA in material and post-mortem contamination. But we said, well, let's just do it anyway. And this is one of the great beauties of having your own my seat. You can just get on and do the experiment. I'm getting ahead of myself here because I've now got to provide you a bit of historical and geographical context. So the samples we were looking at came from a place called Vats in Hungary, which is kind of in the middle of the country in the north. And in 1994, when they were doing some renovation work on this Dominican church in the centre of the town, they found a crypt that had not been disturbed for over 100 years. The crypt contained over 200 bodies and had been used for burials of middle-class families and clerics, Catholic families from that town, for over a century, from 1731 to 1838. And for some reason, when they just opened this crypt up and took out these coffins, they found that many of the individuals had become naturally mummified. So instead of rotting away completely to skeletons, there was still soft tissue there. We still don't quite know why that is. It may be that they were in pine coffins and that pine contains antifungal materials, but we don't know. Anyway, a few years later, around the turn of the millennium, Mark Spiegelman went in to actually sample these soft tissues here using a fibre optic endoscope, went to various mummies. This is Ildiko Pap, who is the Hungarian museum curator involved in the project. And they were able to show that they had prima facie evidence, well, in fact, more than prima facie, they had good molecular evidence that there was tuberculosis in these Hungarian mummies. Perhaps about half of the bodies that they looked at, they found evidence of tuberculosis in them using approaches such as PCR to do that. So we got involved because they said, well, do you think you can get any sequences out of these samples, any genomes or anything like that? And so our first round of analysis, looking using metagenomics on these Hungarian mummies, involved these 3 individuals, 2 of whom are still alive. One is long since dead. So this is Jackie Chan and Martin Sargent, who were in my group at the time, and I'll tell you about this young lady in a moment. So this young lady is Terezia Hausmann, who died at the age of 28 in 1797. And she'd had a chest X-ray done, which didn't show any signs of TB, but she did appear quite wasted, consistent with TB. And microbiological and molecular analyses had suggested that there was good preservation of TB DNA. We had her death record here, so that's one of the interesting things about these mummies, that we had not just the remains, but we also had documentary evidence as to when they lived and when they died, which is quite interesting, quite unique actually. So I said to Jackie, all right, go and do some metagenomics on that. It may or may not work, we'll see what we get. And she did a MySeq run, didn't work particularly well, got 5.5 million reads. But less than 1% of them aligned against the human genome, so it looked like all the human DNA decayed away. But 8% aligned against Mycobacterium tuberculosis, which gave us 32-fold coverage of the genome. So this was one of those things where I just fell off my chair and said, how can this be? That's kind of what you'd get if you were growing the TB in the lab and trying to sequence it directly. But it was one of those things where there was no other explanation. We hadn't cultured any TB, not performed any PCRs, obtained deep and even coverage. It had to be real. Contamination could be ruled out. We couldn't see any way in which that could be the case. And if you map against the reference strain genome, you get this kind of picture here, where it's pretty even, about 30-fold coverage all the way along, with just the occasional one spike up here, which is the 16S locus, parts of which are conserved with other mycobacteria, which presumably in that specimen as well. And then these dropouts, where there's much less coverage. And you can focus in on these regions, and you can see that there are parts of the genome that are missing. And that's characteristic of the way in which TB evolves. You have these so-called regions of difference, where bits of the genome just get lost in a particular lineage. So this actually made quite a lot of sense, and it allowed us to say, well, this is probably belongs to this kind of Harlem lineage, particular lineage of TB. So even though it was 200 years old, we could start to say something about it. But Martin actually was a very smart guy, because he was trying to draw SNP-based phylogenetic trees and not really succeeding and getting rather puzzling results. And then he realised that his pipeline had been excluding anything where there was less than 80% representation of a particular sequence at a particular spot. When you remove that, you can actually start to see that in certain parts of the genome, 50% of the genome was 50% of the reads were represented by one sequence and 50% by the other. So there's actually evidence there was a mixture of 2 different TB genotypes in the same sample. And interestingly, some Spolliger typing, another kind of typing method, had been applied to this family, the Hausmann family, and showed that there were more than one genotype there. So this suggested that there were 2 genotypes in there. And when he looked at coverage plots, he also found similar things. So as I mentioned before, there are parts of the genome where you get, when you align against this H37RV, you get complete dropout. There's no representation there, because there's nothing, that's dropped out. But other parts of the genome, the count drops by about 50%, suggesting that one of the genotypes has got that part of the genome, the other one has got a dropout there, got a deletion there. So that was interesting, showing that basically there was this mixture. And so we wrote that up for the New England Journal as a letter in the New England Journal, and basically we were able to show the relationship between those 2 mummy genotypes and actually a modern genotype from Germany, and actually document the SNPs that were involved in the evolution of this these particular 2 lineages. So we felt quite pleased that we'd managed to get some metagenome out of this mummy. Then we thought, well, let's go and have another look now in more detail and do deeper analysis. This time the individuals involved in the analysis changed somewhat. So Gemma Kay and Jermin Zhao were the ones who did the experimental and the bioinformatics work primarily. And Nick Lohman, you can see there, and Josh Quick, his student, also contributed to some of the sequencing. And here are some of the coffins that were stacked up in the crypt. We ended up sampling a much larger number. In fact, we settled on 8 bodies in the end that we investigated further. And from those 8 bodies, we managed to get 14 different TB genotypes with varying degrees of coverage. So when we went back and did Terezia-Hausmann again, we actually ended up getting 300-fold coverage of one of the genotypes from her. In one other case, we got over nearly 100-fold, over 100-fold coverage. Many others, we were getting just like 1-fold or just 0.5-fold coverage, but nonetheless enough to be able to assign the genotype to a particular sublineage. All the genomes we found came from one particular lineage within TB, this so-called Euro-American lineage. But the thing that was surprising, that was the same story we'd heard with Terezia-Hausmann, but even louder now, that basically mixed infections were very common. This changed my way of thinking. I thought, I just went around with this unquestioned assumption that you catch TB once in life and that's it. But why would you? If you're living in an area where everyone's coughing over everyone, you're all poor and you're all living together, you're going to get coughed over multiple times. So there's no reason why you wouldn't get infected multiple times. We found 5 out of the 8 bodies actually had more than one TB genotype, and one of them had 3 genotypes. So when you think about it, you sort of think, well, actually, the way we do TB microbiology, you pick a single colony usually and follow it up. So if there were multiple genotypes, you wouldn't find out usually, and it's a lot of effort to go and pick multiple colonies. And when I looked in the literature, there was a study from KwaZulu-Natal that said actually 1 in 5 cases in that high prevalence area had mixtures of organisms. And there was one case where 4 distinct genotypes reported from a single patient. So it certainly changed my thinking, but it seems that the mixed infections were actually more likely than not in that time of peak tuberculosis, when tuberculosis was probably at its height in Europe. Then what we did was we drew some phylogenetic trees and used this kind of dark art of Bayesian phylogenetic analysis to do some dating. And we came up with a date of this whole lineage 4 back in the late Roman period, around 400, as the start of when all of this evolved. And this is consistent with a date that had also been put out a few weeks before in another study of about 6,000 years for the whole TB complex. Now, some people have said, well, actually, TB goes back to the Neolithic, and therefore we must be wrong. In fact, some of our co-authors who were working on the project said that, and we had to be very careful in the way in which we phrased what we said. And we are currently working on some samples from a submerged site in Israel called Atlit Yam, and trying to determine whether we can get evidence of TB from the Neolithic. Unfortunately, that's not going quite as well as I'd like, and we're not quite clear whether we've got a signal or contamination in those studies, but the work is still ongoing. And that's being written up. Now, I'm going to run out of time if I don't move fast forward. Now, some of you here who have a medical background will be saying, what are you going on about all this arcane nonsense about 200-year-old dead people? Interestingly, Bill Gates noticed it and tweeted on it, and that gave me bragging rights with my children. Oh, Dad's research being noticed by Bill Gates, someone they'd heard of. But the point is well made that it's a bit late, isn't it, to be making a diagnosis 200 years after the event. And there's this phrase that sometimes put in anatomy dissection rooms, the living will teach the dead. We kind of thought, nobody's actually done this on contemporary TB specimens. Let's see if we can do it. So why don't we get some sputum samples from TB patients? Now, as it happens in England, I don't know if it's like that in Northern Ireland, it's all stitched up. There's kind of mafia of people that run the public health labs, and we couldn't get a look in there. But we went to the Gambia, and we actually managed to get some sputum samples from patients in the Gambia. And my PhD student here, Emma Doughty, did this work. And we were able to, she was able to collect these samples, extract DNA. And what we found is in these 8 samples, we've got different numbers of reads aligning to the human genome, different numbers of reads in terms of yield. But when we aligned against the H37RV genome, we got different, also different amounts of coverage. The best that we got here was 0.7-fold coverage. So we never got like 1-fold coverage. But nonetheless, this is, if you look at the number of bases aligned, even in the worst case, we've got over 30,000 bases aligning against M. tuberculosis. So it's an absolutely clear signal that there was TB present in that sample. So you can use metagenomics to make the diagnosis of TB. And then what we also were able to do, though, was in 7 out of the 8 cases, we were able to use the SNPs that were present in the sequences to align them on a phylogeny of all TB lineages to show where those samples, the genotypes of those samples fitted. So 2 of the genotypes belong to this so-called Africanum, West African lineage. The others belong to the Euro-American lineage. So that was interesting. We wrote that up not too long ago. Now I'm going to give you one last story because I know time's moving on, and then I'll shut up. So I'll take you to the village of Gerudo, which is in Sardinia, in the northwestern part here. It's a medieval village that was abandoned in 1426. And here we work with this lady, Raffaella Bianucci. She's a physical anthropologist, and she had access to material from this site, particularly from a 56-year-old male who had been excavated in 1997, buried in the second half of the 1300s. And here's a picture of the skeletal remains as they're excavated, and around the pelvis, all these funny little nodules, calcified nodules were found. So they were taken out, and we were given one of those nodules to analyse. Now, those of you who have a clinical background, you can go, calcification. What organism causes calcification in this kind of setting? And you'll be thinking, he's going to tell us about TB again. But the great beauty of doing shotgun metagenomics, if we'd have gone in with probes and PCRs and stuff, we'd have just done that and then said, it's negative for TB. But because we went in with an open approach where we just did shotgun metagenomics, what we actually discovered was a whole load of reads aligned against brucella in the sample. Brucella's unusual, it's got 2 chromosomes. You can see we've got this fairly even coverage. That's 0.7-fold coverage in our initial study. Then we got out to 7-fold coverage when we went deeper. And we showed that there were some signatures of ancient DNA damage in the brucella as well. And we were able to actually use the information that we got from the genome, this medieval brucella genome, to place it in a phylogeny of, so it's clearly a Brucella melitensis, and its closest relative was this Melitensis ether, and it sat within I haven't shown it here, but it sat within a clade of Italian isolates. So interestingly, it's like continuity of a clade of Brucella melitensis in the Mediterranean region that spanned for many hundreds of years. And we know that there's been a long history of brucellosis in Italy. Herculaneum, the skeletons from the beach there show signs of this. But there had actually only ever been one other ancient DNA detection of brucella in the past, and nobody had used shotgun metagenomics before. And this organism was caught from sheep and goats, so we keep coming back to these kind of Neolithic inventions. Long history of sheep and goat farming in Sardinia. And we don't know whether this guy was a farmer who actually lived close by sheep and goats, or whether he ate dairy produce and caught it that way. He did have these changes in his skeleton where you get fusion between the vertebrae, which is said to be associated with a sedentary lifestyle, but whether that is evidence that he was really somebody who just ate dairy produce is not really clear. We're going further with this. We've got some medieval leprosy genomes, which we're currently analysing. We've got 3 of those on the go at the moment. I've been to Atapuerca in northern Spain earlier in the year, and we're looking at sheep bones and sediments as well, but mainly sheep bones. And we're looking at a Korean mummy and looking at the gut contents of this Korean mummy at the moment. So we've still got plenty of work to do there. Now, to draw all this together, where might we be leading? Well, the idea that you can just sequence DNA, it's a bit of a simple idea, but it's an idea that seems to have developed legs recently. There's this idea that we could just do macromolecular monitoring, you might call it. We can look at shed DNA, maybe other macromolecules like RNA and proteins that are found in urine, feces, or I gave this talk in America, so I misspelt feces, apologies for that. A lot of people interested in circulating DNA, RNA and proteins, so the people in the cancer unit down the road will be telling you all about their liquid biopsies and diagnosing cancer directly from this. And there are many applications here. So it's interesting in the way in which microbiology may well be converging with all these other different aspects of medical practice. One last thing I'm going to say before I finish. I've spoken mainly about the kind of analysis and the work we've done, but obviously you need a platform to do the work. And one of the things that I've been involved in the last couple of years is setting up this cloud infrastructure for microbial bioinformatics, or CLIME as we call it. And this is an MRC project funded to the tune of 8.4 million. And what we do with this is we will provide you, the users in the academic microbiology community, with bioinformatics resources. We can give you virtual machines that you can log into. They will come pre-compiled with most of the common software that you might use to analyze genomes, to assemble them, to annotate them, and whatever. And we provide training. We've been doing that. We've got a series of research fellows, and we've also refurbished space. But if you're interested in that, you can go to our website. If you want to actually get your own virtual machine, so you can do these kind of analyses, and in fact there is, on our discourse.climb.ac.uk, we've actually got a tutorial on, you can redo that brucellosis analysis if you want to get practice in it. But if you go to brin.climb.ac.uk, you can register. As long as you have a .ac.uk email address, then you can get your own virtual machine and start working on these kind of things. Okay, I've taken you through quite a lot. We've been all Birmingham, Germany, Hungary, Italy, West Africa, into the cloud, and now back to reality. Just leave you with some last thoughts. I first gave a talk about the German outbreak and the metagenomics in Germany, in Berlin, near where the wall was. And this quote was particularly apposite, I think, because it's from an American journalist who went to the Soviet Union in the 1920s. And he came back and said that. Seen the future and it works. And it kind of did work, didn't it? It did work for 50, 70, 60 years, but then it failed. But I think we have seen a glimpse of the future here. These techniques are kind of working, and they are going to start working more. But there's another useful quote here from William Gibson, cyberpunk guy. That, the future's already here. We can do a lot of this stuff. It's just that we haven't got it working consistently out of the research lab into clinical practice. We've got it working in some parts of the UK, but not in others. We've got it working in Europe, but maybe not in Africa. And so there's a lot of work to do just to get these things working. And whether we really will be getting rid of our agar plates and gram stains in the future and just doing everything by sequencing, well, maybe that's what we'll try to do, but whether we'll get there or not, predictions are difficult, especially about the future. And that's me finished. Thank you very much.
Thank you, Mark, for this interesting talk. Floor is open for discussion.